Okay, so yeah, so we're gonna get started on this session on model parameter exploration and optimization. I'm gonna give a brief introduction, and then the idea is to do one of the NetPyNE tutorials on setting up this batch exploration and optimization of parameters using what we call grid search, and then briefly also show an example of using evolutionary algorithms. So I'm gonna, start presenting here. Okay, so I wanted to start just by mentioning the difference between these two concepts. They're kind of related, but they do have some differences. And optimization, we refer to more of at the beginning of the model building process, and it refers to finding model parameters that make the model reproduce usually some experimental data. So this optimization process requires some target where you want the model to get. For example, matching the experimental firing rates that you observe. We can use several methods, like here at the top you see a figure using this grid search or parameter sweep, where you just try many different values, fixed values, or these more dynamic models where, like evolutionary algorithm, where they try to find the model parameters using some more complicated searches, and we're going to see a bit more about those. And exploration refers more to once you have built the model, you want to evaluate the model parameters. So you want to play around with them to either gain some insights of what's going on in the model at the different levels, biophysics, of how, for example, how some of the channels generate oscillations in the network or so on. You want to test some hypotheses, could be some experimental hypotheses that your collaborators have shared with you, or you could want to make predictions. That's one of the main things for models. They should be able to reproduce the data and make predictions. So here's an example from this motor cortex model, where once the model was built, we evaluated the response of the model of this layer 5B population to different levels of motor thalamic input and noradrenaline modulation. And this was guided by experimental data that we had, showing that these two variables were the ones responsible from switching from the quiet behavior of the mouse to the movement behavior. So we wanted to use the model to explore this parameter space. Okay, so we're going to focus on parameter optimization in this talk and examples. So why do we need it? I'm going to start showing some examples of how we can use it in different scenarios, and then an overview of the approaches that we have. And we'll end up focusing a bit more on these evolutionary algorithms. So the idea in parameter optimization, as I mentioned before, is you want to find the optimum parameter values or a set of parameter values that achieve a target. These targets can be different levels of experimental data, from single cell properties like the FI curves to firing rates to actual behavioral tasks. And the models adjusted can also vary a lot depending on the model you're building. So let's see some examples. In this single cell model we were tuning, we wanted to modify basically the biophysics of the cell, so the different channels and some of the biophysical properties, like the capacitance of the neuron, the leak, the axial resistance, and the density of a number of channels, calcium, potassium, sodium. And our targets were several. So in this case, we wanted to match several features of the cell, including the FI curve, so this response of the firing rate in response to current injection, the actual spike shape, so we were trying to reproduce exactly the shape of the different spikes of the voltage traces, and also the subthreshold voltage shape. So when the cell is not firing, we still wanted to match sort of the response of the cell. At the network level, this example is a bit old, but it's still applicable here. So the things we usually modify are the connection weights, but not usually each of the synaptic weights individually. So we find groups of weights, for example, all the connection strengths from excitatory to excitatory connections, all the connection strengths from excitatory to inhibitory, or the strength of the background inputs that provide like a background noise to the network. Okay, so we have like these more global parameters that we find and fine adjust to try to match some targets in the network, which can be, for example, the average firing rates, the coefficients of variation, as we mentioned before. It can be at a more high level, the local field potential spectra, some statistics of the peristimulus time histogram. So there are many things you can match depending on the data you have and the objectives of your model. And notice that we always try to optimize these parameters within biological realistic ranges. So when you do this optimization, you have to define the range of parameters to explore, and that is usually constrained by the literature. And finally, we can have models which actually try to reproduce some behavior. So for example, we built this model of motor cortex connected to a spinal cord model that learned to drive a vertical arm. So this was a very realistic musculoskeletal arm with muscles, tendons, the bones, and so on. The movement was also reproduced by a robotic arm. And what we were trying to do is optimize some what we call meta parameters, some training parameters in the system. For example, parameters of this reinforcement learning algorithm that we were using, the types of exploratory movements, so a set of more high-level parameters, and try to optimize those to ensure that the arm was behaving like the experimental data we had from macaques. So we wanted to reproduce data from monkeys reaching to 4 different directions. And so here in the bottom, you see on the left-hand side, this is the macaque data. So it started in the center and it had to reach 4 different targets. And this is the model results after some of these optimization of parameters and how the arm was able to reach the 4 different targets. Okay, so I wanted to mention quite an important aspect here, which is this idea of parameter degeneracy. Sorry, this doesn't fit here. I forgot to update this. But essentially, the idea is this is a famous set of results from Eve Marder, where she showed that the same network dynamics could be obtained with many different parameter variations. Many different parameter combinations lead to the same results. So this is from the somatogastrium cell in, I don't remember the species exactly, but she had some experimental data, and she found many different combinations of parameters in the model that resulted in the same dynamics here that you can see at the top.
Yeah, and somebody was asking in the last session about how do we know that this is correct? And this is a problem, parameter degeneracy. There's lots of combinations that can produce the same behavior. So you have to make sure that you're within physiological ranges, and there's different checks you can do to sort of minimize this. But yeah, it's a problem.
Yeah, but additionally to this, sorry, how can we know that our model, for example, or our measurements are not missing, in this case, some channel, for example, some missing channel that hasn't been studied before?
Yeah, and that is the complexity of modeling, is that because you're never going to have all the details. So you have to decide which details, aspects you think are the most relevant, and you might want to try different models. What happens if I add this channel to the model? Does it really add anything to the explanation? Is it vital to have it? So all these questions are always interesting to ask. Something interesting that Eve Marder said in the last talk in neuroscience was that this parameter degeneracy is also present in biology. So if you record from different species, you can look at the density of channels, and you can see variations in the order of 3 orders of magnitude, even in different from the same species, different animals from the same species, and you record this concentration of channels, and they produce the same behavior and the same physiology, but the actual channel concentrations can be very, very different. So that means it's not only a problem that happens in modeling, but it's actually present in biology, so it might not be such a problem.
That's really interesting, Salva. I was not aware of that. I guess I missed Eve Marder's last talk, but that's cool. So maybe neurons in biology also solve the same problems with radically different channel distributions.
Exactly.
Cool.
So yes, I'm gonna briefly go over different methods for parameter optimization, and of course the most classical one and that's been used for many years, it's hand tuning, which is doing this sort of manually. So you start with your model, and based on your experience and what you know about the experimental data, you might want to start tweaking some of the parameters gradually and seeing the effect that they have. And this can be very effective, especially if the model is not particularly complicated, there's not too many parameters, and you have a good insight into how the model works or how the system works. However, as soon as the model starts getting more complicated, this approach is usually untractable, and you can spend a lot of time just modifying parameters one by one and not seeing a clear result because there are many dependencies between parameters, for example. So another approach that we use quite a bit, and it can be quite effective, is this idea of brute force or grid search or parameter sweep. They all more or less refer to the same thing, which is basically to sample the parameter space, usually in on a homogeneous fashion. So you have intervals that are fixed for each dimension. And here's a representation on the right-hand side where you have like this grid of parameter values that you want to explore in multiple dimensions. The problem with this one is usually that it grows exponentially. So as soon as you have more than 3 or 4 parameters and you want to explore more than 3 or 4 values for each, you start getting thousands of combinations and they grow very fast. So you have to be very selective about the range of values you want to explore and which parameters are relevant for the model. And that can be also a very hard decisions to make because you don't really know beforehand which ones are the relevant parameters or the interesting values. And then there's a sort of a third category, search algorithms that try to use some algorithm to find the best parameters. And some examples are gradient descent, these are one simulated annealing, or genetic and evolutionary algorithms. So depending on your parameter space and how linear or non-linear it is, some of these may work better or worse. We've been using evolutionary algorithms for some time and they work well. We've recently switched to a new one called Optuna, which is a similar dynamic search they call, which also works very similarly and is a bit more cost efficient. And so I wanted quickly to show you an example of one of these grid searches, the brute force approach that we used. This is, for example, we are seeing the color represents the firing rate of one of the populations, these layer 2 IT cells, in response to a combination of 4 different parameters. So you see this is a 4-dimensional parameter exploration that you can plot in this way. You have 2 parameters here on the small squares, the weight of the background, of the inhibitory background, and at the top and on the horizontal dimension, the weight of the excitatory background. And in the large square, so to speak, we have the rate of this background inhibitory and of the rate of the excitatory. So basically 4 parameters, and we can see how they affect the IT2 rate. So this gives you a good sense of which range of parameters you want to stick to, or a specific set of parameter values that you think would be good for your model. Evolutionary algorithms, of course, everybody knows where they come from genetics. And the idea is they have several steps. So you start with an initial population, and there are several steps, computational steps involved in this, both in biology and in the implementation that we do. So they usually reproduce with each other, which means different parameter combinations or different individuals that they call here. They exchange genes or they exchange parameter values so that you get new individuals. Then you have this component called mutations, which are actually really important to increase the diversity of animals, because otherwise you would always keep the same sort of genes within a population, and you want to introduce these novel variations. And then you make this fitness calculation, so you evaluate which of the individuals are better based on the fitness function that you define, and that is another very important component how you define your fitness function or your objective is going to determine how well your algorithm works. And this is not always easy, especially if you want to measure several things at the same time. So if you want to measure in the cell the response, both the quality of the voltage and how the voltage matches the experimental data, but also this FI curve, then sometimes it's not easy to weight the importance of each component. This is another representation, and a bit more of the same. I'm gonna skip some of these because we are running a bit late. Here, I just wanted to emphasize, that this is a sort of process that you have to refine over time. So you're not gonna hit with the right answer the first time, and at least on our experience, we start with some idea of what we want to do, and then over time we see we should have explored this other range of parameters for this specific parameter, or we had some kind of bug in the model. We many times discover bugs because the results that we get are not what we expected, and so we have to restart the process again. So this is an iterative process, and so we have to analyze the results of our optimizations that we do. We run these large simulations, we get thousands of results, and then we should always analyze and see if we can improve this optimization process. And the ways to improve them are changing the range of parameters explored, changing the actual parameters that we look into, updating or improving the fitness functions, and of course also changing the meta parameters of the algorithm. As I was mentioned, this evolutionary algorithm, for example, has many steps, and you can modify the values of each step. For example, this mutations that happen, you can provide a coefficient. What percentage of mutations do you want? And that can sometimes affect the results a lot. So here I'm just showing this is for the A1 model. We just wanted to see for this particular set of results that we run, many simulations. You see the firing rate for each of the populations in the model. There's like 40 populations. And we see that the color represents the fitness of the solution, so how good the algorithm evaluated that solution was. And we see that for most populations that range within reasonable values. This one here, for example, presented very high values. So maybe we want to look into what's going on with this one and why it's producing such large firing rates. And yeah, this kind of insights that the analysis can provide. So another example is here, looking at one of the parameters that we were optimizing. In this case, the connectivity between the inhibitory and the excitatory for the parvalbumin cell, so one of the inhibitory cell types, and how strongly it projects to other excitatory cells. And we compare that, we do like a correlation with the fitness, and we see that most of the solutions lie within the 0 to 1 range for this parameter. So maybe, and you know, the lowest fitness is also within this range, which means that maybe we can constrain the parameter values explored for this parameter, the range of values between 0 and 1 in the next set of simulations that we run, because it might be that all these values are not really producing any useful solutions. Okay, this is a very vague example, but just to give you the kind of things you can look into. And this is a similar thing, but looking at the firing rate of one of the populations and how it changed with the fitness of the solutions obtained. So, sorry, lower fitness is better because we're looking at fitness error. So that also applies to this plot here. And so we see here that when we get the best fitness models, so the models that best fit our data, the firing rate of this population is kind of slightly high. Perhaps we wanted it to be below 20 Hertz, and here in this case, it's between 25 and 50. So this might be an issue that we may want to look into, why this population is firing so high when in the best solutions that we found, for example. Okay, and I think that's about it. Just a quick overview, just to mention that the package that NetPyNE uses is called Inspire. So this is a Python package, and NetPyNE just uses this under the hood. So it's not really directly accessible to the user. But just so you know that this is a package that's available, you can also use it separately, and it has a lot of different optimization algorithms built into it: genetic, evolutionary, simulated annealing, and a bunch of them. And I won't get into this, but as I mentioned, some of the things you can optimize for to improve the solutions are the meta parameters of the optimization algorithm. And these include things like the population size. So how many individuals, parameter combinations are you evaluating each time? The mutation rate, as I mentioned before, if you include too much mutation, it might take too long to converge. If you have no mutation at all, it might converge too soon without reaching the best solution. So you have to always find a compromise. And yeah, I think we'll leave it there. So I want to try to show now first an example of, yes.
Can I ask you a question?
Yes, of course.
Why are, it's not the first one that I observed, but why is it preferred to use heuristic algorithms instead of deterministic algorithms in terms of optimization?
So deterministic algorithms like gradient descent don't usually work with the types of models we build, because our models tend to have very sort of non-linear chaotic dynamics. So this gradient descent try to go in a sort of very linear fashion, and they usually end up in local minimum, local solutions. So yeah, so we usually algorithms that are more sort of stochastic and search for random spaces in the parameter space work a bit better.
But they have the issue that could not be optimal, right? I mean, the solution that you found maybe is not optimal for the problem.
Yes.
Okay.
Yes, definitely, yes. And here with these very detailed biological models, the issues that we have potentially thousands of parameters. So we try to constrain as many of them based on experimental data and just select a small subset of them to optimize. But the real parameter space is immense.
Okay, so one idea should be first to reduce the amount of parameter, right? Like, yes. I don't know, to do some PCA or some nonlinear fashion to decrease the variability or the parameter number.
Yes, usually we haven't used PCA. It's more of a sort of based on our experience building previous models, for example, or as much as possible, what parameters we know are well constrained by literature, and we want to keep those fixed, and what parameters we know would have a more strong effect on whatever kind of things we are looking at. So, but yeah, there are many approaches, and of course, we're always exploring better solutions to do this.
Thank you.
So I'm going to share the link to the collab, which is the tutorial 8 in the NetPyNE website, and that's the introduction to these parameter optimizations.
I have a quick question.
Yeah.
How long does the whole tuning optimization process usually take?
Years. Now for the A1 model, the last that we're building is actually taking much longer than we expected, because it does have a bit more complexity than previous models. So we have like 43 populations and 4 different inhibitory cell types. So they have like very complex relationships between each other, and it's been quite hard to find good solutions. Also because we have thalamus involved, so it's taken quite a while, and I think we are in at least a year now into the process. We seem to have now a decent solution, but it's been quite hard. And for the M1 model, probably a similar amount of time, but also because, as I mentioned before, we tend to find bugs in the code or things that we missed in terms of the experimental data we use. These are very complex and detailed models, so if you miss a number or something was wrong, then you have to start the process almost from scratch every time, and that has happened a couple times. So I really encourage you to look in detail when you're building the model in the first stages of just putting all the data in, to thoroughly check that everything is right as much as possible before going into these optimizations, because optimizations are quite costly. And yeah, if you had some issue that you didn't realize, you have to start again.
May I ask a question?
Mm-hmm.
Do you usually start from very complex model like this auditory cortex model mentioned before, or do you gradually increase the complexity? For example, start from more like Potjans-Desman column model, then add some simple morphology, and then replace some neurons with more morphologically complex, and so on. So do you use this gradual approach, or you usually start from?
Not really, because we have seen that they don't really scale easily. So the solutions you found for simpler models, the parameters don't really help you that much to build a more detailed one. So it's usually more of a additional work to tune just a simple one. And yeah, for example, the Allen Brain, they published the V1 model. They published 2 versions of it. One with point neurons, this ADEX model, which is like a single point neuron, very fast model, and one with very detailed morphological neurons. And they mentioned in the paper that the parameters from the single model didn't really apply to the more detailed one. So they built both of them just to compare and have a fast one, but they really didn't benefit much from having the simpler version.
Yeah, I see. And I think that's what you expect, because they're very different, but maybe not the exact values of the parameters, but certain intuition about which parameters affect what, and which of them are most useful, for example, like that. So.
Yeah, for sure. I mean, some of the parameter ranges we use are always from previous models, and some of them simpler models. So it's definitely something that can be useful, and we have done in the past. Even from the motor cortex, the mouse motor cortex model, we used a lot of those parameter values and intuitions to build the auditory thalamocortical model.
I see. Okay, thank you.
Okay, thank you. Okay, so we're gonna see how to set up these what we call batch simulations, and we call them batch simply because in this case NetPyNE is taking care of running not one but multiple simulations, and this can be used both for parameter exploration and for parameter optimization. So we're going to start with the simple case of this grid search parameter sweep, but first I wanted to go over the minor changes that you have to do to the files that you already know about. So here in this, if you clone this or make a copy of this Google Colab and you run the first 2 or 3 cells, you should get here on the left-hand side, if you click on the folder, a folder called NetPyNE course 2021. And if you click on that, you will get 5 different files. Okay, so we're going to see the structure of these 5 different files and how they're set up to run this batch. So make sure, I'll give you a couple of seconds just to get to this point so you can also look at the files. Okay, so up to now we have seen most of the models in a single file. The models did have like a structure. Let me maybe get one of the previous examples here. We could see that they had a clear structure in the sense that you had the net params on one side. Okay, so you had this section called the network parameters, where you define all the sort of biological parameters of the network. Then you had a separate component, which was the simulation configuration, which is a different object of a different class, which is specific to each simulation you run and what duration, etc. you want. And the third sort of distinct component was the actual command from the API to run the simulation, to create the network, run the simulation, and analyze the results. So what we're going to do, and the way most models in NetPyNE are structured, is separating these three things into three separate files. We haven't done that until now because they were relatively simple models, so it didn't make much sense. But when you start building more complex models, it makes sense to divide these three things. And that is the way that this tutorial 8 is structured. So we can start with this one called tutorial 8 NetParams. And if you click twice, you should see it popping up here on the right-hand side. So this is essentially the same thing we had. So it's the network parameters, this object, defined in this file and nothing else. So this is it. Okay. We do require a specific line at the top that is different from before, and it's this line here from main import config. Okay. I'm not going to go into the details. It's just like a programmatic thing to be able to run the batch simulations. But remember, if you have issues, remember to check that you do have this line here at the top when you're running batches. The second file would be this config, the CFG, the simulation configuration. And again, we can see that it's just very simple. We define this object, CFG of the type simconfig. Notice that here I'm using CFG, and that is the way we have tended to use it in most of the models, just because simconfig is very long and you use this quite often. So you just make CFG of the type simconfig. And the third file that we have divided into this is this one called the Init, which is the file that we're going to use to run the simulations, to run an individual simulation. Okay, and this has the same command we had before, this create, simulate, analyze, and it has an additional line, this one here, that says, let me see if I can show it completely. Okay, so it calls this method called read command line arguments. Okay, and the reason for this is that when we run these batch simulations, what NetPyNE does is generate a configuration file for each of the parameter combinations we want to explore, and runs each of the simulations calling a command line argument and passing that file in the command line. So you don't really need to understand any of this. It just works internally. But you do need to add this method here in the init.py file. So again, we have these three files, the netparams, the simulation configuration, and the init, which are the same as you had before. And the only new file that we need is this one called tutorial 8 batch. So it's a very simple file, and it's the one we're going to have here in the Google Colab. I'm just going to close this. And this is the file where we're going to define. This is it. This is all you need. Where we're going to define, I can remove this line here at the top. We're going to define the parameters we want to explore, and a bit of the options of how we're going to do this exploration, which method we're going to use, if it's grid search, if it's evolutionary algorithms, whether we're going to run using MPI in our local machine, whether we're going to run on supercomputers, and so on. So there's a bunch of functions that are built into NetPyNE and that you can use. So let me just go over this. This is just the typical structure, but we could even simplify this more, just to keep it as simple as possible. We get it out of the function, and we have the basic commands here. So we're going to create a params variable of this special type in NetPyNE, and this is an ordered dictionary, and it's just convenient to use this for data type to set up the batch. So you just create it always at the beginning here when you set up these batches, and then we fill in this params dictionary with the list of variables we want to explore and the parameter values. Okay, so here comes an important concept, and is that all these parameter values that we want to explore, they have to be previously defined in the config file. So for example, we want to explore the tau, the time constant of one of the synaptic mechanisms. Okay? So we have to define that in the config here. So you see we have this variable config symmectile. We give it an arbitrary initial value. And of course, we have to define what that variable means in terms of our model. So we have to go to NetParams and specify that variable here. Okay, so you see this is where we define the excitatory mechanism in symmecparams. And for the tau2 value, instead of having now a fixed value like 0.5, we pass on this config variable. Okay, so this is sort of 3 nested levels. At the bottom, we have this NetParams, which is the most like stable long-term structure where you define your network model. Above that, we have this simulation configuration, which is something that you can change for each simulation. You might want to try different durations or different parameter values, and you specify those here. And then at the top level, we're going to have this batch where you can modify any of these parameters that are already present in the config file by setting those here in the params structure. So it's always set in the same way. So at the beginning, this can be a bit confusing to wrap your hand around what is this structure in these 3 files, but you will get the hang of it because it's very similar for all the batches that we set up. Okay, so once we have defined the parameter values, the next step is to set up this batch object. So this is something that is a class defined in NetPyNE. It's imported up here. And we just need to define what parameters we want to explore, what is the name of the configuration file, in this case is tut8 config.py, and what is the name of the netparams file, in this case tut8 netparams.py. The final step is setting some sort of run configuration options. So what is the name of the label of this batch that is going to be used for naming the files, the output files, where we want to store this simulation data. So we can set a specific folder for this simulation. The method that we're going to use, in this case, is going to be grid, but you can also set here things like evolve, or we have another one called Optuna. Okay, so you have different methods and some parameters specific to how to run this model, this batch. Sorry. For now, we're always going to use this thing called MPI bulletin, and I will explain where this comes from. But here you would set other methods, like if you were running on supercomputers, you might want to use this thing called SLARM, which is a workload manager, workload scheduler that submits different jobs to the supercomputer, and that is also built into NetPyNE, so you can use it. So MPI bulletin comes from something called the bulletin board, which is a specific feature of the Neuron simulator that allows you to run simulations in parallel and have what they call a master node and several slave nodes. So the master node is going to distribute jobs into the different slave nodes, and each of those
and child nodes, Salva.
Oh yeah. We had to use the new. What was the proper nomenclature?
I believe it's parent and child.
Yes, parent and child. So before we had the other one, but it's not politically correct anymore. So yes.
Let's be woke here.
So the parent and child nodes, and so yeah, the parent distributes the jobs, and the child nodes execute them. Okay, so for this to work, you have to have MPI properly set up with Neuron. And we will help you get that set up next week. Early on Monday, we're going to have a session to set up this locally in your machines. So this magically works actually in the collab. At least yesterday it worked. So we can try to run this and see what is the output and what's going to happen.
Excuse me, a question.
Yes.
Sometimes there is a situation that 2 parameters are functionally dependent. I mean, or you have to do something more complicated. Is there an option, for example, if you have a grid of 2 parameters and you know how to derive the third in a function analytical way, for example, is there a place where I can derive some parameters from the grid point or optimization point where I am?
So
do you understand what I mean?
I think I get what you're saying, yes. And so this kind of setup actually gives you quite a bit of flexibility because you are really defining, let me see if I can open NetParams. So you can do here quite complicated things. So the parameters don't necessarily need to match directly to one of these values, but you could, yes.
So I can, for example, define a variable before this line, which is gonna be a function of several parameters and use it in your network and so on, right?
You're muted, Salva.
Sorry, yeah. So yeah, so you can use these config variables in any way you want. So for example, you can have like a condition here. If config method 1 or and do equal a, then create some kind of population. If it's equal to something else, then do something else. Okay? Or you can have the config variable be something that you use in an expression, a equal b times config variable.
Yeah.
And then this is what you use here. Okay. So yes, this gives you the flexibility to create quite complex dependencies and structures.
Did you talk about yet, or do you want to talk about right now grouped parameters, or?
We
don't have to now, but there is a way to group multiple parameters together because sometimes they vary together. So for a batch, you want to vary them together. Well,
yes.
we'll discuss that later.
Yeah, got it. Thank you.
Okay, so let me show what happened when we run the simulation. So the first thing that you see here is NetPyNE is essentially creating the different configuration files for each of the parameter combinations that we decided to explore. So we had 2 parameters, each with 3 values. So we're going to have 3 by 3, 9 parameter combinations and 9 simulations. And as you can see here, NetPyNE is creating one of these config files stored in JSON format for each of them. We can even go here and have a look at one of these config JSON. And this essentially has all the simulation configuration values with the specific values that we or NetPyNE set for that case. The first one would be 3.0 and 0.05. So if we look at synmec tau, in this case it's 3.0, and con weight is 0.05. If we look at another of these config JSON files, it would have here 0.01. And NetPyNE then uses these configuration files to run the different simulations. So we can also see here the different values that are used in each case, 3 and 0.05, okay, up to the 9 different parameter configurations. And these are going to be stored in the output folder with indices that represent the different values. So 0, 0 is for the first parameter combination, 0, 1 for the second, and so on. So for each of these, NetPyNE is actually running the corresponding simulation, and you can see here the output. Okay, so it's run a simulation, and it got 10 Hertz for the sensory population and 17 for the motor, and it stored it in this file here, 0, 0. It's also plotting some figures, so you can directly see the result here. For the first configuration, we see this was the raster plot. And for the last one, for example, we can compare it and see a different raster plot. Here, the average rate was 8.7 Hertz. And for the first parameter configuration, it was 13.7. Okay, so as you can see, with a simple batch file setting up the different parameters to explore, NetPyNE has taken care of running all the corresponding simulations and saving them here in a nice structure that you can then analyze and plot. Okay, so any questions about this? I know it's a lot at the beginning. We're going to do quite a few examples today. Joe's going to show some examples. Then Joao at the end is also the last 2 hours. You're going to practice different examples with setting different types of batch. So you'll get the hang of it. I heard a question, I think.
Where do you change the hyperparameters of your optimization method?
Yes, so in this case, we're using grid search, so it doesn't really have any hyperparameters. But if we had here evolve, which is the next one we're going to see, you would set those here. Select mutation, whatever. We're going to see an example of that right now.
Cool, thanks.
Any other question? Okay, so let's move on to the evolutionary optimization example, and that one is here. Let me share the link. I'm gonna close this one. So I'm gonna post the link here. So make a copy of that one. Okay, so this one you're gonna see it's a very similar structure. Let me just clone the repo. So the files for these are on some GitHub repos that we created, and the idea is that you just clone them into the Google Colab, and you can use the files there. Okay, so I think we have the necessary file. So as you can see, the structure of the files is very similar to what we had before. So we have this netparams file, which defines the model. Yes, actually, in this one, we have 2 examples. We have like a simple network and a more complex network. I think I'm just gonna, well, doesn't matter. So we have 2 types, so we're just gonna focus on the simple one. So this is the definition of the network parameters. If you select the network type complex, then you would define this more complex one. But yes, so here are the network parameters, and we can see that already there are some here. Down here, we see the ones that are sort of parameterized into the config file. So we have the probability of connection, the weight of the connection, and the delay. So these 3 are the ones we're going to play with. We also have the configuration file, the simulation configuration. Again, we're going to focus on the simple one. And we have the standard parameter, simulation options, and these 3 initial values for these 3 parameters, probability of connection, weight, and delay. And we have the init file, same as before. At the end, we create, simulate, and analyze the simulation. And before, we have this line, which is required for the batch to work. Okay, so same structure as before. And now we have this batch run file, which should really be called just batch.py. And that is what we're going to play with in the collab here. So yes, let's stick to the simple case. So we have 3 parameters, probability, weight, and delay. And in this case, because we're using evolutionary algorithms, this range of values indicate the minimum and the maximum value for each one. So this means that the probability of connection, we're going to explore values that go within 0.01 and 0.5, within this range. For the weight, we're going to explore values between 0.001 and 0.1. And the delay between 1 and 20 milliseconds. Okay, so these are the parameter ranges, which is one of the most complicated things to estimate, and that you can play around quite a lot until you get the right ones. And then the other thing, I'm just going to remove this for simplicity. Just keep the simple one. The other thing we now need is what we called before the fitness function, which is telling NetPyNE how do we know if a solution, if one of the simulations is good or bad. Okay. And for that, here we implemented, not very complicated, but you know, has some complexity to it, which is a way to compare the simulation firing rate of a population to the target firing rate that we want, but within a range. So we use actually a Gaussian function, a normal function, normal distribution, and we want to say that the score that we give that model sort of decays with this normal function around where the mean would be the exact rate that we want, and we can define the width of this normal function, this Gaussian function. So by defining the width, we say how close do we need our solutions to be to that firing rate, or how flexible are we? If we don't mind that the population rates are not exactly, we can increase the width of this Gaussian function, the variation. And if we want them to be very exact, we can make this value smaller. And the third parameter we can define for each population is the minimum firing rate that we want to see. So we have this sort of Gaussian function centered around the value, but we might not want it to go below 0.1 hertz, for example, or below 2 hertz. We can also define it. So the fitness function itself is defined here, and you're going to pass this function as an value of the evolutionary algorithm down here. So yeah, I'm not going to go into the details of the implementation of this Gaussian function comparison, but one important thing is that you have access to this data structure inside the fitness function, this thing called sim data. And if you remember, sim data is where we store the results of the simulation, and so that data structure is passed here so that you can use it in the fitness function, and essentially you can compare your target that you want to obtain. For example, you want to see 15 hertz in one of the populations with the actual simulation data. So for example, here we are comparing the target with sim data pop rate scale. So population rates is something that can be stored by NetPyNE. You can tell NetPyNE to store the average firing rate of each population. It's going to go into this data structure, and you can then use it for the comparison here. And so that is the fitness function, and then we essentially follow the same steps as before. We create this batch object. We pass the parameters. Here we didn't include the netparams file because if you use the default names netparams.py and config.py, you don't need to include it here. And we set up the label of the batch, in this case simple, the folder where we want to store it. The method, notice here, is going to be evolutionary instead of grid. And as before, we set some configuration of how to run this. We're using the same as before, this MPI bulletin board the script name, so the initial file to run is called init.py. And these are just options if you wanted to run on some different setup on an HPC using the SLAM. I'm going to remove them for now. But just so you know, you can include these kind of things in this structure. And finally, so we have this structure called the evolutionary configuration that tells NetPyNE the options, the meta parameters of the evolution. There are several evolutionary algorithms that are built in. We're going to use the custom one that you can customize here with a few values. We pass the fitness function that is defined up here. We pass the fitness function arguments, which are also defined here. And we set the meta parameters. In this case, the population size, which is what we saw before, which is how many individuals, how many of these parameters are we going to try to evaluate on each generation. The number of elites, which is another parameter I'm not going to go into. The mutation rate that I mentioned before. The crossover, which is how they mix together this reproduction. Whether we want to maximize or minimize the function. So do we want to, these fitness function values, we want them to go down or up. Usually it's down because we want to minimize them. And the maximum number of generations. So how long do we want this algorithm to go for? Usually this will be something like 200, 1,000, 5,000, something like that. These are parameters times sleep and max iter weight that are used to, in case some of the simulations fail, how long is NetPyNE supposed to wait until we go on to the next generation. And this can be important if you are, you might have some errors and issues. You don't want to get stuck waiting for those simulations, so you just want to move forward. And the default fitness value, if there is no simulation output, you use this value, which is usually relatively high. Okay. So we can just run this now to see the output of what NetPyNE is going to do. And remember, okay, so I'm gonna undo my changes here because the tabs and spaces got messed up. Okay, so as before, you see that NetPyNE is generating these different config files saved to JSON, and it's trying out different combinations of values. Note these values are now randomly selected within the ranges that we specified before. So up here we said the probability should go between 0.01 and 0.5. And in the first one that NetPyNE ran, the value selected randomly was 0.05. The same for the other parameters. And it generates 6 of those because we set a population size of 6. It then runs those simulations and gets results. So for each of those, you get the firing rate of each of the two populations, the firing rate of the M population and the firing rate of the S population. So what we said is that we want to make the M population be around 15 Hertz and the sensory population be around 5 Hertz. And based on this, NetPyNE will calculate a fitness error value. So for example, here you see the M population was 15.2 Hertz. So that's pretty, what it was, M was 15, so that's pretty good. And the S rate, fitness not really matching. Let me see. I will have to check this, but in theory, the fitness value should be lower when the rate matches what we want to look for. So overall, NetPyNE just gives an overall fitness value to this solution, 6.7, 36.4, and it will try to minimize these values as it goes generation after generation to give at the end a final solution. Okay, it's just showing the first, well, I don't know. In generation 0 here we see, for example, let's go back. It generated 6 evaluations. The worst candidate had a score of 505. The best one had a score of 6.7. And you can see the standard deviation of the different solutions obtained. In the second generation, we expect this to be slightly better. So the best was 6.7 again. So the best is already pretty good. So you might not find a better one. But the overall median and average of the solution is getting lower. So it's getting better. And if we go to the last generation, generation 4, the best solution is still 6.7. And at the end, you should be able to, here it is, best solution was obtained here. And it's basically telling you, well, it should show you the firing rate of the two populations and how close it matched the original target. I'm not sure exactly of the values here, but that is the overall idea of the fitness function and the evolutionary algorithm implementation. Okay, so do you have any questions? Yes.
Hi, Salvador. I ran it here on my computer.
Mm-hmm.
I got a bad solution that is different from yours. Is that related to that kind of degeneracy that we saw?
Yes,
Like
and in this case, I must say that every time you run it, this is not deterministic as before. So this inspired algorithm for the evolutionary algorithms, it actually generates different values, tries different values every time. So you won't get the same as I get here, but statistically should be quite similar. And yes, you should be able to find similar solutions with populations having similar firing rates with different parameter values. And that is exactly the idea of parameter degeneracy that we mentioned. I will look a bit more at this particular example and whether it's working properly or not. But at least the actual execution of the program, you can see how it works. And I will check the values later and give any updates. Any other questions? Okay, so thank you everyone, and we
Salva,
will...
I have a rather naive question. So,
okay.
For example, if I have an action potential with me and I would like to match it with an experimentally generated signal, where does one load that? I mean, is the fitting function doing that kind of fitting, or is it?
Yes, so you would load your experimental data at this stage here. So you see here is where we're putting essentially our target data. In this case, it's very simple. We want the S population to have a firing rate of 5 Hertz. But if you had an action potential, you would load the data, and that would be maybe the different data points of the voltage. And you could do like an Euclidean distance, like point-to-point distance between your simulation voltage trace and the one you have, for example. Or you could extract some features like the peak amplitude, the width of the action potential, these kind of features. You would extract them here, both for your data and from the model, and then compare between them and find a fitness value measure.
I see. Okay, thank you.
Any other questions? By the way, there's an example. I will put the links now in the examples folder of using evolutionary algorithms and grid search, I believe, both for optimizing single neuron models. And I think what it's optimized is the FI curve, the firing rate response to the current injection, using a similar method as here. So you can apply both to the network and to single cells.
Do you recommend having like a multi-tier where you do like first you try and find the neurons and then you try to like create the network by try to find is just like run optimization for each individual
yes.
as you go larger in your network.
So yeah, so we usually definitely do the neurons separately. So we tune each neuron independently to experimental data, and then we tune the network itself. And sometimes we do tune the network, for example, without local connectivity, just with the external background inputs that we want to use to see that the network is at a baseline level. And then we add the local connectivity. So yeah, we tend to do these things gradually. And even for tuning very large networks, a method we have found that works well is adding one layer at a time. So we might start tuning layer 4 until it's has good firing rates, and then we add layer 5. And we keep the weights relevant for layer 4 relatively in very short ranges, and we make the parameter ranges of the layer 5 that we just added very large, and we tune both of them again, and then at layer 6, layer 3, and so on. Any other questions? Okay, so thanks so much, and we'll see you after lunch at 2. Joe's going to show some more examples of this and how to analyze the results. Thank you, everyone.
Thank you.
Thank you. Thanks, Alva.
Thank you.
After lunch.
Thank you.